Okay, thank you very much. I'm delighted to be in this grassroots movement. Two incredibly inspirational persons just spoke before, Aled Edwards, who really started the concept of trying to upscale and upgrade the entire ambition, fitting tremendously well in this target 2035. And Claire, who's been my sort of corporate companion in these years and really a partner in crime. We're having a lot of fun, and I really would like to mention here again that we are incredibly proud of what Pfizer has been doing, both with the vaccine, but also with this protease inhibitor. And I think that it is very, very nice that Claire is here with us, because she will continue to take us to drug things that we haven't been dragging so much yet. So I would like to start by saying that my view of a biological system, like a cell being a fundamental unit, is that you are really assembling, if you want, chemistry from the environment into this entropic island of a biological system, and that is a highly organized little entity that is clearly requiring to selectively take up from the environment the kind of chemical components it requires. And you can think in big terms, from the nutrients to the building blocks, to the ions, to whatever it takes to make life go around. And obviously, major issues are the provision of energy, but also the ability to create mass and multiply that. And transporters are exactly at the interface between, if you want, this environmental chemical world and this biological little citadel of a cell, of a biological unit. And you can take any cell across the different kingdoms of life, and you will have the same problem. How do I selectively take up what it needs? Or at least, how do I be sure that I have what I need? And so membranes are there to delineate, if you want, a biological system, and they give, if you want, they protect the genetic information, but also they protect the metabolic environment and identity that allows a biological system to be different from the environment. The concentration of the solutes that are within the cell, but also within the subcellular compartments in the case of eukaryotes, is very, very important, not only because you're creating the concentration that are driving reactions, but also because you are creating energy by acquiring different concentration that you can then exploit, such as in the chemiosmotic hypothesis of ATP generation across membranes. So it's all about differential accumulation, and it's all about being sure that the right reactants are at the right place at the right time. And what is absolutely clear, if you think about it, is that everything has to be connected in many ways. First, we know that every charge needs to have a countercharge. We know that every, if you want, energy expenditures needs to be accounted for. So it's boring to think about a cell as an accounting system, but it is in fact an accounting system. You need to be economical in what you're doing, and everything needs to be accounted for. So that is very important in a way that you say, therefore, the activity of these transporters that remain quite elusive in many sort of ways, as we will see, are fundamental because they represent the gates to the system, to the differential accumulation of reactants, and they have to be regulated also because we are convinced that this is not happening in a random fashion. And I won't have the time to go through the different type of transport that we know through membranes. There is passive diffusion, there's ionophores, there are channels where you really, for a short time, make a perfect connection between the outside and the inside. But then there are transporters, and there are some that requires ATP. They typically belong to the ABC family. And we are in this talk and in this consortium that Claire mentioned, more focused on the so-called SLCs. We'll see what they are. And they are not primarily active. They don't use ATP directly, but they can either be facilitative, and they let concentration that exists be facilitated in their, if you want, equalizing that by allowing a better transfer across a membrane, or they actually are active, but they're secondary active. They use a gradient that was built in another setting to drive against a gradient. So it so turns out that it is we think, the largest class of transporters in the human genome. There are 456 genes and counting, divided in many families. They are integral proteins, as was already mentioned by Claire, and they are both at the cellular surface, but also at the, across different subcellular compartments. They are a superfamily. It's difficult to know exactly the folds that we'll talk about this in a second. And they have different transport mechanisms. So in a way, they're not a family. They really are an operational group. And they have at least 3 different, probably more, mechanisms of transport, and they are associated with quite ancestral kind of folds that are conserved in evolution. And so bacteria are using similar system. And this nomenclature that you follow is SSC, stands for cellular carriers. And then you have a number that stands for the family. Then you have a letter that stands for the subfamily. And you have then the individual members, such as SSC5A2 is the second member of the A subfamily of family 5. And so you know it probably as SLG2, a target of liphoxins, and very important glucose transporters. But you probably know, for example, you need SSC6A4, which is the 4th member of the A subfamily of the 6th family of the SLCs, and that is a serotonin transporter and the target of serotonin uptake inhibitors. So you are familiar with this. And recently, because of AlphaFold, we looked, or Evandro Ferrada resolution looked at how SLC behaved, and it's clear that there probably have been misallocated. So some of the subfamilies probably need to merge, and others may represent different folds. And so there are quite a few folds on top of the 5 major that we already knew. So there is quite a lot of heterogeneity, which is actually one of the reasons why they have not been so easily targeted. And so some time ago, Dave Hepworth, Elliot Edwards, Matthias Hediger, and I and other friends wrote a review that reviewed the status of knowledge then. Then there were only a few SLC that were targeted. The list is not much longer, but it will get soon, we think, longer. So there's only about 22 such SLC that were targeted. But as Claire mentioned, one of the peculiarities about this target class is also involved in ADME, because it is, in fact, sort of granting access of biological system to drugs as well, which is a quite unique situation. And there are many, many SLC that are associated with disease genes. And these are just, if you want, OMIM, Mendelian genes, but there are many more sort of situations where disease are associated with SLC. So it's a prominent and important, and if you're not convinced by all of this, you can, in a fairly speculative and totally populistic kind of view, you can say that alcohol addition, skin and hair color, and happiness may be associated with SLC as well. It's not entirely scientific, but it gives you a sense of the kind of things that you can expect if a superfamily is also involved in transporting pigments or transporting neurotransmitters and so on and so forth. And so for some time ago, and this was the origin that I mentioned, we issued, let's say, a plea to the community and say, let's all work on SLCs because they're cool. And if you want, the key of our research strategy can be summarized in this triangle where we say, if metabolism is influencing transporters, and if metabolism is influencing cell state, then it follows that transporters and cell state also have something together. In other words, we think that ultimately you should be able to describe the cellular state of growth and differentiation of a cell by if you want, the group of transporters that are expressed and their activity, sort of if you want, the metabolic exchange program, the chemistry access algorithm behind a particular cell are likely to be defining that cell as much as any other thing. And so what we would like to do is to crack, if you want, the code that is not really known about what does each of the transporters actually transport. And at the same time, we would like to establish a relationship between the different entities that I just described, so that we are able to sort of understand, if you want, the effect of the expression or the activity of each transporters. And then we would like to consider these transporters as druggable and therefore be able to modulate them both in the target 2035 vision, but also in general terms of saying being able to modulate cellular phenotypes and therefore organ and probably organismic phenotypes by modulating access. And here I'm going to be very ultra fast. It just summarizes the fact that it is a challenge to consider SLCs a drug target, mainly because there are membrane proteins. Many of them are actually intracellular membranes, so they're not easily accessible from the outside. The variety of chemical ligands is enormous. You go from an ion to fairly large complex organic molecules and peptides. So it's clear that you won't have one common kind of pocket of, let's say, chemical binding, which is an additional level of difficulty. It's also true that it's not easy to catch the exact window of activity of an SLC that you're able to monitor in an assay. They don't have, or they may have, but they don't have a common second messenger that you can use to monitor activity. And we don't have really easily applicable methods. And for those SLC that are electrogenic, meaning that their activity are changing locally the membrane potential of a cell, there are some common ways we can do it, but it's only a fraction. And Resolute and Resolution are these IMI public partnerships that involve all the people that are talking today and are clearly sort of there to reach out to the community with just a platform, if you want, to maximize interaction among the community. We would like to create the reagents, the data sets, the assays that are going to be lowering the threshold for as many people as possible to start working on these proteins, particularly as targets of chemical agents. But of course, our ambition goes even beyond that, because we would like medical people to start thinking about SLCs and so on and so forth, even more than they're already doing. And one of the things we're doing here is a picture that we took recently at the Resolute Consortium at Novartis in Basel. Not everybody was there, and those that were not there were there in spirit, because we have a picture of them. I'm on top of the stairs. I'm holding the picture of my hero, Alan Edwards. It's not perfectly visible, but those who know will see it. And we really sort of, one of the things we started, and this was an idea of Dave Hepworth, we say, why don't we review what we currently know about assays, particularly cell-based assays, to see whether we will be able to create an impact and improve the number and the quality of the assays that will be available to target. And this is really something that is very concrete in terms of enabling the community. And so one of the things that we're doing, say, in an academic sense, not that the other one is not academic, but in my own laboratory, let's say, is to try to say, why don't we try to see where SLCs are involved in cellular processes and why? And the other question we're trying to address is, how is function integrated? So in a way, a sort of an academic background kind of activity. And one of the things that has been very useful, even if there are new versions of this library, was a CRISPR-focused library that Enrico Girardi designed and executed, that was allowing us to not look at all the genomes in the genome, genes in the genome, but just the SLCs, meaning that we could do many more genetic screens for SLC involvement in a very efficient, even multiplexed way. And we've done this now for quite some time, and I can tell you that wherever we look, there is an involvement of SLC. For example, Enrico himself and his colleagues were able to show that there is a big involvement or a more significant involvement than that probably many people anticipated between SLC and drug uptake, meaning that more drugs that are currently characterized as requiring an SLC require an SLC to pass membrane. We did a lot of interaction proteomics already, let's say, in early days. One of the things we identified together with Boehringer Ingelheim was, for example, a protein associated with SLC15F4, which is located in endolysosomes, important for innate immunity, for the response to nucleic acid and other challenges. And here we found that an SLC was actually providing a platform for a protein called TASL to come in and then lead to the phosphorylation of IRF5, an important finding. And recently we published a paper that shows that surface located nucleoside transporters are involved in modulating the activity of chromatin remodeling proteins such as BRD4, just as a way to show that, where there is metabolism, there are transporters involved, and where transporters involved, you can assign particular cellular function that are depending on the limiting activity of these transporters. And I will briefly mention the fact that we have been in the vision of trying to target this, let's say, target group, developed some PROTACs that work on this. And we also, of course, are trying to deorphanize them by using metabolomics and ionomics. This is something we do mainly within Resolute, but also a gigantic effort that was still interpreting led to the understanding that one of the things we could do to try to decipher the function of an individual gene was to see whether it would be synthetically lethal with another gene. And so making essentially combination of SLC loss of function mutations and trying to see what would be synthetic lethal with what, and trying to imply that there must be a functional, let's say, link between two SLC that are simultaneously required. And now going to the chemical biological approach, it was really sort of the heroic effort of Ariel Ben-Simon, was to say first he checked whether in principle several past transmembrane proteins would be amenable to this targeted protein degradation that is induced by IMiD-like ligands that sort of are mediating the interaction with cerebron and then with the degradation machinery on the proteasome. And it turned out that independently of where the SLC was located, yes, it could be degraded, except for those SLC that are located at the inner mitochondrial membrane, understandably perhaps, but it was very nice to be able to confirm that. And that paved the way for a design of an SLC that was targeting SLC9 family, SLC9A family members, that actually was sort of a hybrid between different compounds that were known to inhibit this family of SLCs and the mentioned moiety that binds to cerebron through a linker. And we had to fiddle around with a proper linker, but then we created, or Ariel created, indeed, an efficient PROTAC that was very good at degrading certain family members, 9A1 as one, but also a few others. And in depending on the cell type, there are different kind of spectra of SLC9 that are degraded. We are still trying to understand with genetics and proteomics how clean these are. We suspect that they're not as clean as we wish, but they are very potent, and they do seem to have a propensity for certain cell types. And so this is something that we are trying to understand, the ability to inhibit GSPT1, which is a well-known translation termination factor that is often associated with, let's say, off-target activity of the so-called IMiDs, does not account for the toxicity. So we are still trying to understand where this potent toxicity comes from. But the most important point here is that through this work of Ariel, it becomes clear that one can try to use this approach for targeting SLCs. And I will briefly mention another route before we finish, which is exploiting exactly the synthetic lethality map that I just mentioned. And that is the fact, using the fact that two MCT1, so monocarboxylate transporters, SLC13A1 and A3, are synthetic lethal to each other, at least in this cell type that we used. And so if you are inactivating one, you don't kill the cell. If you inactivate the other one, you don't kill the cell. But if you inactivate both, you do. And that was the basis for Wojtek Dvorak, a student in my lab, PhD student in my lab, to think maybe we can design a screen that is perfectly isogenic, using isogenic cell lines, each one having a particular genetic makeup that will allow to direct the screen, the chemical screen, in an intelligent way. And so this is what is done using the panels of cells that were either mutated in one or the other SLC or not at all. And if you use this cell, you can see that SLC1, famous inhibitor from AstraZeneca, is indeed only killing those cells that do not have an SLC6 in A3, so they are then specific for A1, and vice versa. You could, using syngopine, which is not a very specific, but still a quite SLC certain A3 specific sort of compound. If you're mutating the 1, then it becomes dependent on A3, and then if you inhibit A3, you kill the cell. And by doing this, using 90,000 compounds, he was able to identify 1,000 compounds that then he was sort of very nicely able to restrict to those that had exactly the desired characteristic of only killing SSC16A1 knockout cells, so they were dependent on SSC16A3 and on no other SSC16 family member, because he can, in fact, and I show you briefly here, reconstitute the missing gene also with other orthologs like SSC16A7 or A8, and show that the drugs or the compounds that he identified were specific for A3, because they would not be inhibiting a cell that had only A7 or A8 expressed. And this is sort of a paradigm of what you can do if you don't know anything. I'm not going to go into, if you don't know anything about your SLC, you can either try to make a product out of it or for it, or you can try, for example, to do this genetic dependency and exploit this. And the vision that we have in the lab, when we are good-mooded, is to say, if one think about these transporters as they are, conveying chemical matter across the biosphere from plants and bacteria and animals in the food chain, we are going to be able to understand, if you want, each individual organism in the ecosystem as a temporary chemical assembly that is governed by the ability of taking up what is important for that organism, for that period of the life of the organism. And in this vision, the environment acquires a different importance, because we may be in a molecular medicine kind of era, but we are in a molecular medicine era embedded into an environmental chemical process. And we dress up as SLCs to meditate of what it is like to be such a protein. And we have the ambition of saying, if we were to crack this code, would we be able to do many things? And there are many questions that include xenobiotes, that include how we can influence a sort of cell activity by different cocktails. We have the aspiration of creating these tool sets, including data and including assays that will unlock the target for the community. And we clearly think that the ability of modulating these metabolic states through governing the different gates will be very important. And we clearly, and this is my timer, sorry, and we clearly would like to sort of do this. And there are a number of people to thank. I will just mention, and we'll mention Enrico and Kai-Chun and Ariel, because they were important, but the Resolute team was fundamental. And in our part of the organization, Tabea Wittmer is the person that is mostly concerned with keeping everybody sort of aligned. Thank you very much. This is a fantastic initiative, and I wish Target 2035 or possible success.
Let's get this shared. All right, hopefully that's good.
Yep.
Okay, great. So yeah, so I'm going to talk about our work studying using structures, binders, and small molecules in targeting and characterizing SLC transporters. And honestly, Giulio's introduction and preceding me was a pretty perfect starting point, just because not only is he world-class in this field, but he gives the very big picture ideas of what are we talking about, what are we trying to address. I mean, he's even talking about the biosphere and doing multiplex experiments, versus I am very much a structural biologist, biochemist, thinking about single proteins, single molecules, even single amino acids of those proteins. And so where I start is from a perspective like this, of the SLC transporters carry out some function to move something across the membrane. They can be importers, exporters, exchangers, or they can even carry information. These are the transceptors that signal the presence or absence of a molecule. But I mean, as Giulio mentioned, there's 400 and something of these transporters, and because there's so many and they're involved in so many different processes, they're obviously then often mutated in disease. So of those 400, about 20% have a known genetic mutation associated with disease. And those are spread across a number of transporters with a number of different substrates, and that's therefore a number of different biochemical pathways and physiological pathways. And then honestly, quite a variety of phenotypes and severity of phenotype. But in terms of the druggability and the targeting of SLCs, they're remarkably underrepresented, as Julia mentioned. There's more than 400 of them, and which is similar on scale to say GPCRs or kinases or proteases, and even more than the number of ion channels. And yet, if you look at the percentage of drug targets, a percentage of drugs that target SLCs, it's disproportionately small. And a lot of this has to do with the points that Julia made about how they're undercharacterized and to some degree difficult to work with. And so I want to particularly point out some of the things that we need to know in order to drug these SLCs. And one particular example that I'm fond of is the SLC13A5 protein. This is a citrate transporter that's known to be mutated in a seizure disorder. And what we can see is looking at the mutations that are known. The wild type protein traffics very nicely to the plasma membrane. One particular pathogenic mutation clearly does not. It's somehow arrested by the protein quality control machinery and never makes it to the membrane. But then a second pathogenic mutation, this T227M, does make it to the plasma membrane. So this one's curious because you can imagine it doesn't make it to the membrane. Okay, you don't have the activity you need, but this one makes it to the membrane, but still non-functional. So that tells you that even within a single protein, a gene, you can get very different defects in terms of the protein activity, and that means that you would need two different methods of treating this, two different types of drugs in order to resolve this disease. And so that was just one particular protein where it actually is somewhat well characterized. But as Julia mentioned, many of these are undercharacterized proteins. Some of them are completely uncharacterized proteins. And so a lot of the work that we're doing is to try to generate tools to study these SLC proteins. And we're generating two different classes of molecules that are actually fundamentally trying to do very similar things. We're generating protein binders. These can be nanobodies or nanobody-like proteins that bind our target SLC with very high affinity and specificity, or we're also generating small molecules that target again a particular SLC and modulate its activity. And these both have very distinct value, but both valuable in studying SLC transporters. And so to give an example of how the binders are useful in terms of studying SLCs, well, there's the obvious things of doing immunohistochemistry or immunofluorescence to look at the localization of a particular SLC, either in tissue or even in individual cells. My favorite thing to do with them is to try to use them to solve structures, in that these SLCs are often very compact membrane, well, they're all membrane proteins, sometimes they're very compact. And so there's not a lot of surface for making crystal contacts, and there's not a lot of protein outside of a detergent micelle or the membrane for trying to generate a cryo-EM reconstruction. And so often having a high affinity binder is invaluable to resolving both of those issues to solve a structure. Additionally, these binders can modulate the function of an SLC. So that's both useful scientifically, if you want to do a particular experiment, maybe lock a transporter in a particular conformation, but you can also imagine how this would be extremely valuable in terms of developing biologics to target these SLCs. And so the way we're going about generating and validating these binders, and this is our work within Resolute, is that we express and purify these SLC proteins. And this is extremely non-trivial. This takes a lot of work from some very talented postdocs and technicians, where we need to get it to be homogeneous, and stable. And many of these SLCs they don't express very well. When we're purifying them, we're taking them out of the native membrane environment. So we have to add a detergent or some other condition in order to protect the transmembrane helices from the hydrophilic and aqueous environment. And some SLCs don't tolerate that very well. So it takes a lot of work to try to optimize this. Once we get it optimized, we send it off to collaborators who are very talented at generating these protein binders to our SLCs. This can either be through direct immunization of llamas to generate nanobodies, or using more in vitro methods such as phage display libraries. But ultimately, they give us a collection of protein of sequences that code for nanobodies that bind our SLCs, and then we express and purify them, and then validate them through a series of experiments, including something like immunofluorescence. This is the work that's done by others in our work package in Resolute. But so then once we have these nanobodies and they're validated, these are explicitly what we're going to provide to the community through the Resolute portal. In terms of using small molecules as probes for SLCs, well, hopefully you know the value, but just to reinforce it, these can be great biochemically because it's a way to modulate the protein's activity on a single cell or even in some sort of synthetic system. You can also study the activity of the protein physiologically in an organism. So in this case, it's treating a mouse with a particular SLC inhibitor. And we can see that in this case, it's a decrease of citrate incorporation into hepatic lipids. Lipids. And so now you can see how we can use these same chemical probes both on a very small scale and even in an organism to understand physiology. And then finally, again, my favorite technique of structure, often having a high affinity small molecule binder is invaluable for either stabilizing the protein such that you can solve the structure, or capturing a specific state in the reaction cycle, or maybe just the binder itself is medically interesting. Often you'll get structures in complex with a drug and you want to understand, okay, how does the drug work? How can I make the drug better? And so when we're working to identify new SLC chemical probes, and primarily we work using in vitro and in vivo assays. And this is our work, sorry, this is right in the way. This is our work within EUB Open, but we have some very specific standards that we need to meet for a small molecule we identify to be a chemical probe. It needs to be potent. It needs to act with nanomolar affinity. It needs to be selective among the family members. We want to make sure we're targeting, say, SLC13A5 rather than A2 or A3. It needs to work within the cell. It needs to be soluble and not cytotoxic. And then I think this is especially important. We need a negative control compound, something that is chemically very closely related, but doesn't have the expected activity, such that when you start adding this to cells, or even you starting adding this to whole organisms, you can be sure the effect you're seeing is because of targeting to your specific SLC and not some other off-target effect. And so we're screening for and characterizing these small molecules using sort of two different sets of experiments. One are just fundamental binding and affinity assays. I'm showing here FSEC-TS using thermal stability to measure the affinity of a small molecule to a particular transporter. Or we can also do transport assays, either using the purified protein that we may have reconstituted into proteoliposomes, or we can use a transport assay in vivo using for example, the knockout and overexpressing cell lines that Julia mentioned that are being generated in another Resolute work package. And then to assay those transport experiments, we can use either, say, fluorescence assay or measuring the activity using electrophysiology if the transporter happens to be electrogenic. We can also even do radioactivity. That's sort of the worst case scenario, but that's always an option as well. And then finally, I want to mention one additional way to identify small molecule inhibitors of SLCs, and that's to do it in silico. And this is the work, at least the work I'm highlighting here, is the work of Avner Schlesinger and Claire Colas, where they can take a structure of an SLC or a homology model of a particular SLC, and then in silico, they can dock in a small molecule and then evaluate how well it binds. And in fact, they can do this for more molecule. They can do this for hundreds of thousands of molecules. And so you can see how this would be far more efficient than any high throughput screen you could build. And then they can give you a list of, okay, here are 20 compounds that are likely to bind and inhibit your SLC. And they've had very good success doing this. And so with the time remaining, I want to sort of bring all these points together, show how binders and small molecules and structure can be useful for understanding disease and then potentially designing therapeutics to treat disease. And I want to focus on this disease of SLC13A5. This is, it codes for a protein NACT, a sodium-coupled citrate transporter. So about 7 years ago, there were reports of individuals with mutations in this gene, loss of function mutations. And what that ended up being, the disease being named is SLC13A5 epilepsy. Or the more formal name is early onset epileptic encephalopathy. And what that means in practice is that these are children who have seizures within the first day or two after birth. These seizures are pharmacologically resistant. The children go on to have developmental delays, and they also have a tooth phenotype. And so what we and others want to do, I'm starting from the biochemistry, is want to connect how do mutations in this gene lead to changes in protein biochemistry, ultimately leads to aberration in physiology, and then to disease in these individuals. And so we began on this project, and I want to highlight that when I say we, this was my postdoctoral work with Daneng Wang, and it was a highly collaborative effort from several members of the lab, and then other outside collaborators that I'll mention as we go. But we began on this project through a structure of a protein called VCND, which is a bacterial homologue of the SLC13 family. And in fact, we solved this before the pathogenic mutations were even known. And so this was the work of Romina Mancuso, and she found that VCND, and by inference then the SLC13s in general, are obligate dimers with a central scaffold domain and two peripheral transport domains. And you can tell by how I've named the transport domain that we believe we know what the function is, and that we can see the substrate binding site. In fact, and also the sodium ions bound in a cleft of the transport domain. And from that structure and from some biochemical evidence, we and others found that, or inferred that the family operate under what's called an elevator mechanism, which Julie referred to earlier, where you start in some outward-facing apostate, it binds sodium, it binds substrate, it transitions to an inward-facing state, substrate comes off, sodium comes off, and then the apoprotein recycles back to the outward-facing state. But then that leaves some obvious questions. First question is, well, we have an atomic model of what the inward-facing state looks like, but we don't have equivalent experimental evidence for an outward-facing state. There was a computational model with some biochemical results to support it, but we didn't have nearly the level of resolution and experimental detail for an outward-facing state. And then probably more interesting to this audience is how do mutations cause disease? Because if we map those missense mutations within NACT onto that homology model, or onto the VCND structure, what we see is they're decorated all over the place. There's no single hotspot that would explain why these missense mutations are causing disease. And so we want to understand what's going on. And then additionally, there were several small molecule inhibitors that target NACT, and we wanted to understand how they work and what makes them specific or non-specific among the SLC13s. In particular, I want to point out this compound from Pfizer and this compound from Boehringer Ingelheim, which are highly specific nanomolar IC50s against the citrate transporter, but very weakly or more or less absent inhibition against the dicarboxylate transporters. And particularly interesting, if you compare what we call the PF2 compound from Pfizer to these other compounds, the only differences are in this bulky moiety on the molecules, and yet this one is highly specific for NACT versus these two are relatively non-selective between the citrate transporter and the dicarboxylate transporters. So what underlies the specificity of PF2 and the BI compound? And so bringing in these tools that we've developed and that I've talked about, we generated binders. This was the work of, the binder generation was from the work of Shohei Kawaide and Akiko Kawaide. They generated binders against VCND, and so we were able to solve a particularly useful structure of the protein. And this structure, in conjunction with several others, we were able to capture multiple states of the family. In this case, we captured an outward state using a different homolog. And when we compare the two structures, we can see that the transport domain undergoes an 11 angstrom translation and a 35-degree rotation as it goes from the outward-facing state to the inward-facing state. So that gives us some very fundamental biophysics of how the protein operates. And this will ultimately be important for explaining the disease and the activity of the inhibitors, as we'll see in a moment. But we took that outward-facing structure and we also then, well, let's see. There we go. We also then collaborated with Imad Torshek and his very talented graduate student Noah Trebesh to model this transport. And we see that the transporter goes from out to in carrying the substrate, and then the succinate this is ultimately productive because the succinate is released into the cytoplasm. Okay, and then finally, building off of that, then we wanted to go back and answer that second question of how do the mutations cause disease? And in fact, there already was a classification system for these mutations. There were type 1 mutants. These are what was called insufficient protein. Effectively, the protein never made it, either functional protein never made it to the plasma membrane. And then there are type 2 mutations that there was wild type like levels of protein at the plasma membrane, but you still didn't see sufficient NACT citrate transport activity to support to alleviate disease. But again, mapping these onto the structure, we really couldn't quite explain what was going on with if we simply use this two class system. And so using our structure and using our knowledge of the biochemistry, we were able to further subdivide these mutation types into different classes. And this ultimately is very informative in terms of trying to design therapeutics, in that we now have type 1A, which are early termination or frame shift mutations. And there's not a lot we can do with small molecules. This probably would need a gene therapy. We have type 1B mutations. These are shown in red here. And these we think are a folding defect, because if we look at where they're located within the structure, they're in the core of the protein, either in the core of the scaffold domain or of the transport domain. And so we would affect mutations here to simply to alter protein folding such that it never escapes the ER. It's held back by the quality control machinery of the cell. And so to drug this effectively, now you would need a folding corrector. Whereas, so the type 2s, remember, they make it to the plasma membrane, but they're non-functional. We could then further classify the type 2A. These are mutations in blue, that I'm showing in blue here, that are near the substrate or sodium binding sites. And we expect that changes here would simply alter the ability of sodium or substrate to bind. And so to try to resolve that, you would have to try to find some surrogate substrate that could be carried by the transporter. The type 2B, I actually think, are the most interesting, because these lie at parts of the protein we would expect to be dynamic, either hinge regions of the protein or the surface of the scaffold or transport domains as they slide against each other. And so you can imagine mutations here at these locations would lock the transporter in one particular conformation or another. And now you need some sort of activating molecule to restore the dynamics of the transport domain such that it can carry substrate. And then finally, we were looking at, we actually solved an additional structure of NACT bound to that PF2 Pfizer inhibitor. And what we found is that it actually binds with the carboxylate moieties in the substrate binding cleft, almost equivalent to how the native substrate would bind. But that large, bulky group that comes off fits within a cleft of the scaffold domain. And so this suggested to us two possible mechanisms of inhibition that, to be clear, are not mutually exclusive. One is that you've got this large, bulky molecule that's simply a steric block for the transport domain to move up and down. The second is that with this molecule bound with reasonably high affinity, the sodiums can never be released. And so effectively, this protein is locked in a single state of the reaction cycle, the inward-facing sodium-bound substrate, or in this case, inhibitor-bound state. And then from this, we can even build further and explain the specificity of that inhibitor, in that if we look at how the bulky moiety of the PF2 molecule interacts with the scaffold domain. We see in particular this residue glycine 409. And this residue is an asparagine in the dicarboxylate transporters. And you can easily imagine how introducing a larger side chain would prevent the PF2 inhibitor from binding. And in fact, if we make a equivalent mutation, glycine 409 to glutamine, we see that the ability of PF2 to inhibit the transporter is dramatically reduced. And this is the work of Joe Mendel and his talented postdoc, Jacob Hilton. And then we went one step further and speculated that we see, when we look at the surface where that PF2 molecule binds to NACT, we see that it's very rugged. And so we suspect that you can make fairly modest modifications to this inhibitor and generate something with higher specificity or higher affinity for NACT. And so finally, I need to acknowledge everybody on this. We've got a very talented Resolute team at the CMD in Oxford. We have a very talented EUB open team. So these are generating the binders, these are generating the small molecules, and they're all part of the membrane protein group at Oxford. And then the SLC13 work was carried out, was my postdoc work with Daneng Wang, and it was a very large collaboration. All the structural work requires access to some very expensive resources. And so we're grateful for access. And then we've been very lucky to have funding for all of this. So with that, I guess I'll take any questions.